Okay, so this podcast is on eukaryote gene regulation. In order to go into that, we're going to review a little bit on transcription and translation. So just a reminder that when DNA makes RNA, that's called transcription, and when RNA makes a protein, that is called translation. So when we're talking about description, we're talking about DNA, the DNA language of the genes, being transcribed into mRNA, which is an RNA nucleic acid language. When cells do transcription, they are making mRNA, messenger RNA. There is a template strand and a coding strand. The template strand is the strand that's going to be transcribed, and you can see on the diagram where that one is. The other strand is the coding strand, and that is going to be the same sequence as the RNA, but it's untranscribed DNA strand. There's a bubble that occurs when transcription is occurring, and we're making the complementary RNA strand from the template strand. There is an enzyme involved, and that's RNA polymerase. Actually, there are RNA polymerases, 3 enzymes that are involved. RNA polymerase 1 only transcribes rRNA genes, so that's involved with making the ribosomes. RNA polymerase 2 is the one that actually makes the mRNA. That one's highlighted because that's the one that we talk about when we talk about transcription. mRNA or RNA polymerase 3 is the one that makes the tRNA genes. So each one has a specific promoter sequence that it recognizes. So we know which kind of protein that will be made eventually. Okay, so let's talk a little bit more about this promoter region. The promoter region, as you can see on the strand right here of DNA, it's a region of where there's going to be a binding site for RNA polymerase. Also what's binding there is transcription factors. This promoter is called the TATA box, and that's the bonding site. And you can see it's called TATA because it goes TA in the beginning of the gene sequence. For the promoter box, there is an enhancer region and a silence region. The enhancer region is the binding site for activators. Activators, remember, are going to activate the genes. And then there's a silent region where there's going to be a binding site for repressors, and those are going to turn the genes off. Okay, so what are transcription factors? They're little proteins, and they bind to the DNA, and they basically can turn on transcription or turn off transcription. They trigger the binding of the RNA polymerase to DNA. Okay, so when you're looking at DNA, and you're looking at eukaryotes, their genes have a lot of, they call it junk. They have genes in there that don't code for anything. So the genes are not continuous. There's like spaces in between that mean nothing. So the exons, they're called, are the actual genes that code for the DNA. The DNA actually codes for a real gene. They're the ones that are going to be expressed. The introns are the junk. They don't code for anything, and so they're the in-between sequence, and those are going to come out. So after transcription, what happens to the mRNA is it needs to get edited or spliced. It needs a lot of work after transcription. So this primary transcript that's made, which is we're going to call as the pre-mRNA, is going to get spliced. And so basically what that does is it's going to take out the introns. So all the introns get taken out, and now we have a mature mRNA transcript. It has to be really careful that the splicing is accurate. We don't want any mutations. One little tiny mistake, single base, will add or throw off the whole reading frame, and it causes mutations. The enzyme that's doing this is called SNRP, SNRNPs. It's a small nuclear RNA, and it forms a spliceosome. And so several of these SNRNPs or SNRPs come together and form the spliceosome. These things bond and they recognize the splice site or the introns, and they basically cut it and paste it back together. So then there's more processing that occurs. After mRNA is matured, it needs to go from the nucleus to the cytoplasm to be translated. And so there's enzymes in the cytoplasm that are going to attack this mRNA. So it needs to be protected. So they're going to add, the cell is going to add at the 5' end, a GTP cap. It's a chemically modified molecule of GTP. And so it protects the mRNA from being digested by these enzymes. They're called ribonucleases. And then also at the 3' end, we're going to add about 100 to 300 adenines to that end, and that allows mRNA to be more stable. All right, so the last part is after we have our mRNA all edited out and everything's good, then it goes to the translation. So we're going to go from a nucleic acid language to the amino acid language. Okay, we should have already learned about the code and know how to read the first base, second base, third base on the codons. The start codon is always AUG and the stop codons are UGA, UAA, and UAG. So just as a review, this is tRNA structure. It's a clover leaf. The anticodon is on the clover leaf end and the amino acid attaches at the 3' end. So when tRNA is going to bond to mRNA, it requires an enzyme. It's a really long name, aminoacyl tRNA synthase. And so this enzyme bonds tRNA to the, it bonds the amino acid to the tRNA. That bond requires energy. The bond is very stable so that amino acid can come off easily and make the protein. Okay, this is just showing the building of the polypeptide where you have the initiation, the elongation, and then the termination. And then once we have a protein made, it can go and get secreted, go to the nucleus, go to the mitochondria, chloroplast, all these different places that the protein can go to after that. So this is just a cool little puzzle. If you put everything out there, can you actually tell the story of how it starts from the DNA and makes it all the way to a protein?